All right, guys, so let's begin with today's seminar. So, our this week's topic is the respiratory system. So, first, let's talk about the division of the respiratory system. It's divided into the conducting portion as well as the respiratory portion. So, first, let's talk about the main functions. Main functions include oxygenation of the blood, conducts air to the respiratory portion, cleans, warms, and moistens the air for optimal gas exchange, as well as provides a smell function. So it helps you smell things. And when we talk about the respiratory portion, the conducting portion is divided into two pathways. So we have upper and lower airways. So the upper airways, they include the nasal cavity, vestibule, paranasal sinuses, and your nasopharynx. The lower ones include the larynx, trachea, bronchi, bronchioles, and terminal bronchioles. When we talk about the respiratory portion, this is mainly where the gas exchange takes place. So this includes the respiratory bronchioles, alveolar ducts, as well as alveoli. When we talk about the nasal cavity, so I will explain with the fingerland later after I tell you guys some theory about it. So it is divided by the septum and it is bound to the nasal ala. The outer surface of the nasal ala, it is made up of epidermis and dense connective tissue. So the epidermis would be the stratified squamous keratinized epithelium, and it includes sebaceous glands as well as eccrine sweat glands, and it also has the hair follicles, adipocytes, blood vessels, nerves, and skeletal nasal muscles. When we talk about the inner surface, it is similar to the outer surface, but it also has vibrissae, also called nose hairs, which are for air cleaning, and it has hyaline cartilage for support. When we talk about the wall, so the mucosa is made up of the respiratory epithelium, which is just the ciliated pseudostratified columnar epithelium. So from now on forward, we'll just call it respiratory epithelium, but you guys need to know it's ciliated pseudostratified columnar epithelium. When we talk about the lamina propria mucosa, it has similar content in every organism. So it has blood vessels, nerves, lymphatic tissues, as well as salivary glands. When we talk about it, it also has rich capillary network in the concave for warming air. When we talk about the olfactory mucosa, which is again for the smelling function, it is found on the nasal cavity roof and it contains sensory cells for smell. So there's not much to understand about this slide, more about like, just identifying the structures. So you can just, as we look at the fingerland, this is labeled in your fingerland. So the main things that you have to identify just to know what slide it is. First, you can see that this is the hyaline cartilage. And how do you know it is the hyaline cartilage? Because it has a glassy appearance and you can see the isogenous groups as well. Another thing would be the core of the skeletal muscle. So here you can see it is a skeletal muscle because it has the flattened nuclei which are present at the base. So this whole region is the skeletal muscle. And then when you talk about this area, these are all sebaceous glands, and you can see how many, like they're present in a huge number. So you know this is nasal ala. And when we talk about the epithelium, here you have the stratified squamous thinly keratinized epithelium here. And how do you know it's thinly keratinized? Because you can see this thin layer of dead cells here. So this is just the main diagnosis of the nasal ala. When we talk about the paranasal sinuses, there's no specific slide for it, but you need to know some basic information. So when we talk about it, it is found in small cavities in following bones, includes maxillary bone, frontal bone, ethmoidal bone, as well as sphenoid bone. The tunica mucosa is again composed of the respiratory epithelium, and the lamina propria mucosa includes loose connective tissue, lymphatic vessels, as blood vessels, nerves, and seromucous glands, as I said before. Now, the main function of these paranasal sinuses modulate the voice as well as reduce bone weight. So here you can see all of these are labeled. Here's the frontal sinus near the frontal bone. You have the maxillary sinus, ethmoid sinus, and the sphenoid sinus here. Now, when we talk about the nasopharynx, again, lined by the respiratory epithelium, ciliated pseudostratified columnar epithelium, lamina propria, again, same composition, mixed salivary glands, lymphatic tissue. And when we talk about the transition zone, so when we talk about the nasopharynx, the lining epithelium of this respiratory epithelium, it changes to stratified columnar epithelium at the border between nasopharynx and the pharynx. So this is the transitional epithelium, and the pharynx itself is then lined with the stratified squamous non-keratinized epithelium. Okay, next one which we'll talk about is the larynx, the structure of the larynx. So it is made up of hyaline as well as elastic cartilage plates, and it is supported by the outer layer of loose connective tissue, again with blood vessels, nerves, and adipocytes or fat cells. And the outer layer is called the tunica adventitia, which basically helps support and fix the larynx in its original position. Now, when you talk about the tunica mucosa, again lined with the respiratory epithelium, and the main distinguishing feature is that it contains vocal cords, which are covered by stratified squamous non-keratinized epithelium, and it includes elastic vocal ligaments with lamina propria mucosa. So there is no slide for you guys to identify for the larynx, so you just need to memorize this theory. So when we talk about the muscle, it has skeletal muscles which run parallel to control the rima glottidis, which is the gap between the vocal cords, and this is important for the voice production. So now the epiglottis, it guards the entrance to the larynx, so it prevents the food from entering the airway and result in difficulties in breathing. Now we talk about the epiglottis. Epiglottis it is made up of elastic cartilage. So just how I used to memorize it is epiglottis starts with E and it has elastic cartilage. So EE. So it is made up of elastic cartilage at its core and which gives its flexibility and is covered by thin mucosa. So it has two different sides. It has lingual side and laryngeal side. The lingual side, it faces the tongue and it is lined with the stratified squamous non-keratinized epithelium. And again, the lamina propria mucosa, loose connective tissue, blood vessels, lymphatic tissue. When we talk about the laryngeal side, it faces the larynx and it is lined by stratified squamous non-keratinized epithelium near the top, which changes to the respiratory epithelium at the bottom. When we talk about the key features, again, just for you guys to diagnose the slide, elastic cartilage plate and salivary glands from both sides, which open into the epiglottis for lubrication. So when we look at the slide for this structure, just a second. Yeah, here. So as you guys can see, this is the elastic cartilage core. You can epiglottis, EE, elastic cartilage. And when we talk about different types of epithelium, here you can see the stratified squamous non-keratinized epithelium. So this would be the lingual side. And when we talk about the transition zone, here you can see the respiratory epithelium, which is ciliated to the stratified columnar epithelium. Sometimes under this microscope, it's like hard to identify, but here it's easy because you can see here these strands are represent cilia. So this is the respiratory epithelium. And then there's no cilia present here. So this would be the stratified columnar epithelium or the transitional epithelium. And here you can also see some seromucous glands, and this would represent the duct of these seromucous glands. So I guess that's about it for the epiglottis.
Do you guys have any questions? No? Okay, so we're just going to move. So now we're going to talk about the lung, or we're going to dive into the lung. As you guys know, we have the tracheal tree, which starts with the trachea, and then we're going to our bronchus. So we have our main bronchus that's going to separate into our right and left lobes. And from each main bronchus, you're then going to have your lobar bronchus, because you know that the lungs are divided into lobes. And then we're going to have our segmental, which is for each segment of the lung. So as you guys already know, try to remember, we're cutting these cross-sectional sections, right? So if we're taking a cut like this, we're going to see the alveoli, just in the lung, the alveoli, and we're also going to see the lobar bronchus. Or if we were to take a cut here, we would see the alveoli and we would see some segmental bronchus. And that's what your lung slide is going to look like. Riya will go into a bit more detail about it, but I'm just going to show you guys, just so you guys can understand what I'm going to talk about, because then you guys might be confused. This is what your lung slide is going to look like. And imagine that they took a cut through here, right? So all of this gray part, which is like kind of the alveoli, this is going to be all of this very huge white spaces. And then you're going to see some structures that are going to look different, which are going to be either like the main bronchus, the lobar bronchus or the segmental bronchus. I'm not sure if you guys have a slide that, if I remember correctly, you could see the main bronchus. So we're going to talk about the intrapulmonary bronchi. The word being intrapulmonary means we're going to talk about the bronchi that are inside the lung that you're going to see in your lung slide. So we can have your secondary, which we talked about, which is your lobar, is dividing the lungs into lobes, and you have your segmental or tertiary for each segment. Now, there isn't actually much differences in them. However, in theory, you guys could say that the lumen tends to narrow, and that's why the thickness of the wall layers is reduced. To like, we know that it's going to be covered by respiratory epithelium, and you guys are going to have a smooth muscle layer, which is going to be circularly oriented, and it's going to be around the whole lumen. And it's important that you guys know that it's going to be around the whole lumen because sometimes with other bronchi, it's differentiated. Some are going to be longitudinally oriented, and it's not going to be covering the whole, it's not going to be around the whole lumen. Tunica submucosa, you're going to have loose connective tissue, vessels, nerves, and you're going to see maybe some mixed salivary glands at the end in the tertiary bronchi or segmental. Now, again, this is mostly theory that you guys are going to have to talk about because you guys are not going to be able to kind of see a lot of these things. You're going to see a hyaline cartilage plate around the whole lumen, and that is going to be interconnected with dense connective tissue. And tunica adventitia, again, almost as always, loose connective tissue with accompanied vessels, nerves, and adipocytes. Now, bronchioles. So the bronchioles are structures of lumens that have a diameter that's smaller than 1 millimeter. So basically, as I explained, these are bronchioles. And when we cut them, when we see them in a cross section, we're going to see kind of these circles that are very small in diameter, which would be very different from this because you can see a thick layer. So bronchioles, they tend to have more elastic fiber and they have a lot more smooth muscle compared with the amount of cartilage. You do not have any mixed salivary glands, and so tunica submucosa is also missing. The other layers of the wall of the bronchioles are present, but and they're similar, however, the thickness tends to be reduced. Now, remember, we can see cartilage and muscle tissue in the same region of the wall of the intrapulmonary bronchi and bronchioles. And we're going down a tree, guys, right? So we're going from the biggest to the smallest, and we're going to end with our terminal bronchioles. These are the smallest structures that you guys are going to see, and these are also very noticeable because they look very different from the other bronchioles. They have this kind of star stellate shaped, and there will be no cartilage and no seromucous glands. So the tunica mucosa, you've got, again, respiratory epithelium, ciliated simple columnar epithelium with club cells. You're going to have loose connective tissue, and again, it's going to have stellate shaped appearance. And this is due to the postmortem shortening of the muscle tissue in the tunica mucosa and absence of cartilage. So because there is no cartilage, it's able to gather a specific shape, which is the stellate shape. And you guys will see no goblet cells. And then again, tunica muscularis, you're gonna have spirally arranged smooth muscle layer, which is gonna be very thick, and then tunica adventitia, loose connective tissue. As you guys can see, they're very noticeable, very, very noticeable by their stellate shaped. We have one here, here. And sometimes with the very big ones, you guys are going to be able to kind of tell apart all the different layers. However, again, most of the time, it's going to be just the theory that you guys are gonna have to know. And lastly, we're going to go to the pulmonary lobule. So we ended the bronchial, right? We're talking about our bronchial tree. We ended it with our terminal bronchi, right? Because we're going from main bronchus to lobar to segmental and to terminal, to like the end portion. And in the terminal, we're going to see or find what's known as the pulmonary lobule. And the pulmonary lobule is a morphological unit. And basically, this is going to show us the transition from the conducting or ventilation part of our respiratory tree um, to the respiratory part, if that makes sense. So the terminal bronchial is the end part of the ventilating function, if that makes sense. After the terminal bronchial, we now have the start of the respiratory portion. And this is basically the job, or what does this is the, again, respiratory bronchioles and alveolar ducts, alveolar saccs, and alveoli, which Riya will talk a little bit more in detail about. But you guys have this image in your lectures, and I think it's a very good image to kind of explain what or how the pulmonary lobule looks like. And so this is how it looks like. And you guys can see this is the terminal bronchi, right? Obviously, it doesn't, from this angle, it doesn't have that stellate shaped. You guys know that you only are going to see that in a cross-sectional look. But at the end, after the terminal bronchial, you're going to have the respiratory portion, which is when you have the alveoli. And essentially, in cross section, remember that because of the angle that it's cut, everything is going to look like it's in the same place, but it's not. Okay, it's terminal bronchial, then alveoli, and before the terminal bronchial, you have all the other bronchi. So remember that just because this is how it looks like here doesn't mean that they're in the same level. Um, and yeah, do you guys have any questions before we go into the respiratory part? So just quickly recapitulating, what I just spoke about is all the different types. So again, lung, you have the bronchi. And we just spoke about how depending on what type of bronchi we're looking at, it's going to have a different structure, whether it's going to have mucus glands or how the smooth muscle is oriented. And these are all the ventilating portion. So when we breathe in, the air is going through these bronchi into the alveoli, so then the oxygen can be exchanged for CO2 in the alveoli. You have the alveoli and the capillary, and they're going to exchange. And to get to that alveoli, it's good. Our oxygen is going to travel through all of this bronchi. So the bronchi are going to be the conducting or ventilating part where the air is passing through. The actual respiration, which is the exchange of O2 and CO2, happens in the alveoli. And right now, we just spoke about histologically how all these different bronchi are going to look, which you would be able to see here. Um, so, yeah, alveoli, bronchi, and alveoli, bronchi. And that's it.
Okay, so I'll be talking about the respiratory part of the respiratory system. So the first structures which arise here are the respiratory bronchioles, and they arise when the terminal bronchioles further branch into them, and since they are the first elements that participate in the gas exchange, they are the first. Structures who have the presence of alveoli in them. So the tunica mucosa it is made up of the ciliated cuboidal epithelium, and it also has a few club cells. Right below the tunica mucosa, we have a layer of smooth muscle cells and elastic connective tissue. The tunica adventitia is made up of the loose connective tissue. These are further divided into alveolar ducts and they carry out into alveolar saccules. So, the alveolar ducts and saccules have many similar characteristics. Both of them are going to have the simple squamous epithelium, which consists of the alveolar cells as well as the macrophages. It also has loose connective tissue, which is made up of collagen, reticular, as well as elastic fibers. So, all three fibers are going to be present in the loose connective tissue. And it also has the key difference between these two is that the presence of smooth muscle. So only the alveolar ducts are going to have circularly arranged smooth muscle, and there is not going to be any smooth muscle present in the alveolar saccules. This slide is very, very important because it talks about the two different types of alveoli which we have. So there's the type 1 and the type 2. The type 1 is going to be most mostly present in our body. It's like almost 97%, as you can see here. And it has very flattened simple squamous epithelium, and they have really long processes which are interconnected by tight junctions. So since they are interconnected by tight junctions, the main function of the type 1 alveolar cells is the participation in blood air barrier. Now, type 2 alveolar cells, they have round or cuboidal shaped secretory cells. They are not very frequent, they're only 3%, and they are present like scattered among the type 1 alveolar cells. These are also interconnected by tight junctions. So, since they're interconnected, we also know that they will also somewhat help in the participation in the blood air barrier. So, apart from that, they have a very important function, which is the secretion of a fluid called as a surfactant, which is made up of phospholipids. So the function of this fluid is basically prevention of the alveolar collapse. Just a second. Yeah, sorry about that. Now let's talk about the wall of the alveoli. So it consists of the type 1 and the type 2 alveolar cells. And along with that, it also has a basement membrane, loose connective tissue, which has all the other fibers, especially elastic fibers. That is very important. It also has other cells such as fibroblasts and macrophages. And it also has a lot of capillaries. There are these special type of cells which are called as the dust cells, which are present in the lumen of the alveoli, and their function is basically to compensate for the absence of the cilia. Between two alveoli, there is a very thin space which is called as the interalveolar septum, and it consists of like the two layers of the alveolar cells, their epitheliums, and between here in the middle, there are going to be these pulmonary capillaries, which basically bring the blood over there for it to get like carbon dioxide to go out and oxygen to come in. Yeah, alveolar macrophages. So they are the dust cells, and like the name suggests, dust. So they're going to be helping in the phagocytosis of all the dust particles which are present over there. They also help in the cleaning of the alveoli, the saccules, and the ducts. And since they have phagocytosis as their main function, they're obviously going to have lysosomes because we know that lysosomes are the main structures, the main organelles which are responsible for phagocytosis. Blood air barrier. So there are three main layers which form the blood air barrier. The first layer, which is going to be formed of thin processes of the alveolar type 1 cells, like we talked about before, they're connected by tight junctions. The second layer is formed by the fused basal laminae of the type 1 cells with the endothelium, which is located right below them. The third layer is the continuous endothelium of the capillaries. And since the endothelium is continuous, nothing is going to seep out from the capillaries because they're also connected by tight junctions. So both type of the alveolar cells are connected by tight junctions, which helps prevent of diffusion of the tissue fluid into the alveoli. So that basically they protect the alveoli from having any fluid in them. The barrier is a part of the alveolar wall, and it is located only in the wall of the alveolar capillaries and nowhere else. So it's only going to be present in the wall of the alveolar capillaries. There are these special type of structures which you can call neuroepithelial bodies, and their main function is contraction of the smooth muscle, and they are only present in the bifurcation, the tracheal bifurcation, where it uh, like splits into further systems. This is the last part of the seminar, and this is basically just an overview of the lung slide. And Katrina already explained most of it to us, but just to go over it, it has a spongy appearance because it has a lot of alveoli. So all of these white structures, which you can see on the slide, like these empty spaces, they are all made up of the alveoli. So all of these little lines over here, they are going to be the alveoli, and these are the empty spaces between them. The lung has we know that it is covered by two types of pleura, right? From anatomy we know that, and the one which is directly in contact with the lung is going to be a vesical pleura. So all the structure over here which you can see, this is going to be the mesothelium of the vesical pleura, and this vesical pleura is lying on a connective tissue layer which is made up of collagen and elastic fibers. Does anyone have any questions? All right, then that's it for today's seminar, everyone. Hope you guys understood everything, and we look forward to seeing you again next time.